Okay, so far we have talked about homeostasis as being a state in which a variable, such as blood pressure, is maintained relatively constant. We haven't really talked much about how that occurs, though. So to maintain homeostasis, the human body monitors certain key functions, such as blood pressure, blood glucose concentration, that must stay within a particular operating range. These important regulated variables are kept within their acceptable or normal range by physiological control mechanisms that kick in if the variable ever strays too far from its set point or optimal value. So all control systems have three components. You have to have an input signal, you have to have an integrating center that integrates incoming information and initiates an appropriate response, and finally you need to have an output signal that creates a response. So in essence, this integrating signal will take this input signal and compare this input signal with what's called a set point, this optimal value. And then the difference, often there'll be a difference between the input signal and the set point, leads to the integrating center essentially making a decision as to what sort of output signal should be generated. So we'll go into that in more detail next. Okay, so first of all, let's talk a little bit about forms of control. There's local control. This is the simplest form of control. In local control, a relatively isolated change occurs in tissue. A nearby cell or group of cells senses a change in their immediate vicinity and responds, usually releasing a chemical. This response is restricted to the region where the change took place, hence the term local control. A classic example of this is a decrease in oxygen concentration in tissue leads to the relaxation of muscles in the wall of a blood vessel, causing an increase in diameter of the blood vessel. This leads to an increase in blood flow, helping to increase the concentration. So this would be a type of homeostatic mechanism that would help to maintain the oxygen tissue levels at an appropriate value. And this occurs a lot in your body. There's sort of a misconception that everything's controlled top down, but in fact a lot of things happen via local control. For example, if you're exercising and you're walking upstairs, there'll be very specific muscles that are activated as you're walking upstairs compared to, say, walking just on a level ground. And so certain muscles will be activated more, and as they work, they use up a lot of oxygen. And so you then have local control mechanisms leading to the increased blood flow to those particular muscles that you're using for a particular type of exercise that you're doing. And so this is very important. It's a kind of a fundamental concept in physiology, is that yes, there are certain top-down control mechanisms, and we'll talk about those. But a lot of control also occurs from the bottom up. Locally, tissue is in need of oxygen, glucose, and so forth. And so there's changes that occur locally to help lead to the appropriate response. So local control is very effective for causing changes in specific organs. For example, increased blood flow to leg muscles while biking. However, it's not effective for causing systemic changes. That is, changes throughout the body. Systemic changes require long-distance signaling. Any long-distance signaling pathway that uses the nervous system, any long-distance pathway uses the nervous system, the endocrine system, or both. And these are called reflexive control. Okay, let's go into a little more detail into how a homeostatic mechanism would work. A reflex can be broken down to two parts, a response loop and a feedback loop. So we'll use a simple example using an aquarium. We have a stimulus, which is the water temperature. It's picked up by a sensor, so there's a thermometer that picks up the water temperature. The thermometer sends a signal to the control box, which would correspond to the integrating signal center. Now this integrating center would have a set point. And so this input signal, which is the current temperature, is compared to the set point, and then based on the difference, the integrating signal then sends the appropriate output signal. So for example, if the actual temperature is lower than the set point, then the integrating signal would send out a voltage signal, for example, to the heater, which would turn on and generate heat. And so that would be the response. The target is the heater. This is, in biology, we also call this the effector. So often in biology, the effector or target would be something like a muscle or a gland. And then you get a response. In this case, we're heating up the water. So this is, so far we've talked about the response loop, but now we have a feedback loop. This response feeds back onto the original stimulus, which is the water temperature. So we're increasing the water temperature. The sensor picks that up, sends that signal to the integrating center, and now the difference between the set point and the input signal is not as great, and so it might send a weaker signal out to the target. So less heating would occur at the target. And so this is essentially what's called a negative feedback loop. You have a set point, and what this circuit attempts to do is to minimize the difference between the stimulus, which is the water temperature, and the set point. And so that's referred to as negative feedback, because you're reducing essentially this error, which is the difference between the set point and the input signal. Okay, so I think I've talked about most of these things. There's this idea of a regulated variable, which is very important in physiology. Now, there is a lot of confusion about this, so I should emphasize this next point. Note that in order for a variable to be considered a regulated variable, it must have a sensor. So blood pressure is a regulated variable because blood pressure is directly monitored by the body. Heart rate is not a regulated variable because it's not directly monitored by the body. Okay, so we'll be talking mostly about regulated variables, things like blood pressure, blood glucose levels. Okay, in the aquarium, in this example figure I just gave you, what keeps the heater from sending the temperature up to, say, 50 degrees centigrade? So the answer is this feedback loop, which the response feeds back to influence the input portion of the pathway. So in the aquarium example, turning on the heater increases the temperature of the water. The sensor continuously monitors the temperature and sends the information to the control box. The temperature warms up to the maximal acceptable value, which would be around the set point, and then the control box shuts off the heater, thus ending the reflex response. Okay, so these negative feedback loops are considered to be homeostatic. For most reflexes, feedback loops are in fact homeostatic, utilizing negative feedback. That is, these are designed to keep a system at or near a set point, so the regulated variable is relatively stable. One thing to note, though, is that it's not possible for these feedback systems to perfectly regulate the variable. There are always small oscillations of the value that's regulated, or regulated variable around the set point. So for example, here, let's go back to our idea of having a temperature-controlled aquarium. Now, if you had a very sensitive temperature monitor, you'd see that the temperature actually oscillates around the set point. The set point is 30 degrees Celsius, yet the temperature will sometimes drop below. So this occurs, there's a little bit of a lag. The temperature drops below, the integrating center then detects this change, and then it turns on the heater. Now the temperature actually overshoots a little bit. At some point the temperature is so high that the heater doesn't come on at all, and now the temperature drops down again. And so it'll sort of oscillate back and forth a little bit around the set point. And this occurs, of course, in electronic circuits, but it also does occur to some degree in biological circuits. So a pathway which the response opposes or removes a signal is known as a negative feedback system, which we just talked about. These negative feedback loops stabilize the regulated variable and thus aid the system in maintaining homeostasis. In the aquarium example, the heater warms the water, the response, and removes the stimulus, the low water temperature. So that's shown here, negative feedback. The response counteracts the stimulus, shutting off the response loop. Now there is another type of feedback that's used quite a lot in biological systems, that's positive feedback. The response reinforces the stimulus, sending the variable farther from the set point. So you have an initial stimulus, and you get a response, and that actually increases, say, the stimulus, and then this feeds back onto the response, causing an even greater increase in stimulus. So basically, in this sense, there is no true set point. There might have been a normal value, but once you start to deviate from this normal value, then it's sort of an explosive change, in which the stimulus would get continue to, say, increase or decrease. It would get further and further away from the normal value. And so in these positive feedback loops, you have to have an outside factor that's then turns off the feedback loop. I think this will be clearer when we talk about an example. Here's a positive feedback loop. As I said, the response reinforces the stimulus rather than decreasing it. In the positive feedback loop, the response sends the regulated variable even further from the normal value. This initiates a vicious cycle of ever-increasing response. Note that there must be a factor that leads to the shutoff of the positive feedback loop. So the classic example of a positive feedback loop involves the control of uterine contractions during childbirth. When the baby is ready to deliver, the head of the infant pushes down on the cervix. That pressure leads to the activation of neurons, sensory neurons. These send signals and sensory signals to the brain that cause the release of the hormone oxytocin, which causes the uterine muscles to contract very forcibly. This pushes the head of the infant against the cervix. This increased stretch causes the activation of the sensory neurons in the cervix. They send a sensory signal to the brain, which causes even more oxytocin release, which causes even more contractions. And so the cycle continues and continues, and the contractions get stronger and stronger until eventually the baby's delivered. At that point, there's no more stretch on the cervix, and this stops the positive feedback loop. And so there's a natural endpoint to this loop. Okay, there's another type of system that's very important in biology, which is called feed-forward control. This is a response that allows the body to anticipate a change that's about to occur. One of the most classic ones that's easy to understand is the feed-forward control of the salivation reflex. So in this case, we have a dog. It sees some meat, or it smells it, and even the thought of food is enough to start its mouth watering in expectation of eating the food. This reflex extends even further because the same stimuli can start the secretion of hydrochloric acid as the stomach anticipates the food on its way. So that's another type of control in which it's an anticipatory control. Okay, I think that's it for these feedback loops. Here are some review questions you can check out at the end of the chapter. All the answers are in Appendix A of your textbook.